In this video, we will be looking at fluid movement in cells by doing an egg demo. Have you ever hard-boiled an egg, then peeled off the outer shell to find a thin membrane underneath? This inner membrane is a selectively permeable membrane. It allows for osmosis, the movement of water into and out of the egg. It does not allow solutes to pass through. In order to use an egg in this demo, first we have to remove the outer shell. To do this, we will place it in vinegar. This will remove the outer shell, leaving the inner membrane intact. So what will happen to an egg when it's placed in distilled water? Here we see two eggs which have both had most of their outer shells removed, so that only the selectively permeable inner membrane remains. The egg on the left has not been placed in any solution. The egg on the right has been placed in distilled water. What do you notice about the size of the egg in the distilled water? How can you explain this? Keep in mind, distilled water is pure water with no solutes in it. What happens to the egg if it's placed in a solution that has a high concentration of solutes? The egg on the right is in corn syrup. Corn syrup has a very high solute concentration compared to the egg. What do you notice about the size of the egg, and how can you explain this? In summary, here we have all three eggs. Be sure you can explain what is happening in the situation where the egg is placed in distilled water, a solution with a low concentration of solutes, and when it is placed in corn syrup, a solution with high concentration of solutes. Note, the picture on the left is the egg in vinegar. This is a control. This is what the egg looked like before it was put in the distilled water and before it was put in the corn syrup.